So, hello everyone. Today we'll discuss a corona vaccine like Covaxin, Covishield, and Sputnik-V. What is the difference between how this vaccine works? We have already discussed one of the videos like in DNA vaccine in previously. So now we'll discuss the different ways of how these vaccines work and which one is the better option for preventing the mutant strain of the coronavirus. Now let's start. Now first is you will see. So what is vaccine? So vaccine is an antigenic preparation used to stimulate active immunity and to create an immunological logic memory to prevent the effect of infection. So what is vaccine? Vaccine is an antigenic preparation. What is antigenic preparation? Antigenic preparation meaning any foreign particle which can generate antibody in our immune system. So vaccine is an antigenic preparation used to stimulate active immunity to create an immunogenic memory to prevent the effect of infection. Now the modern vaccines, most of the vaccines now available in the market for coronavirus are either RNA vaccines or inactive pathogen. So we should know about what is DNA and RNA vaccine. So DNA and RNA vaccine is a technique for protecting against disease by injection with genetically engineered plasmid containing the DNA or RNA sequence encoding the antigens against which an immune response is sought. So cells directly produce the antigen causing a protective immunological response. That means here we actually engineered the plasmid containing the foreign DNA sequence or RNA sequence that will generate mRNA or proteins, and these proteins actually acts as an antigen, right? Now we'll see that at previously available the vaccines like live attenuated vaccines, diseases like measles, mumps, polio, rubella, tuberculosis, varicella, yellow fever, like inactive or kill vaccine, cholera, influenza, hepatitis A, polio. These are all we have discussed previously in our another videos. So you can watch that videos. DNA vaccine. So toxoid is an inactive exotoxin is in an active exotoxin like diphtheria, tetanus, subunit vaccine. Inactive or inactive vaccines like in case of hepatitis B, pertussis, inactive exotoxin. Like now other type of vaccine like DNA vaccines and vector vaccine. Now we'll see the vaccines available for coronavirus. Now vaccines are like covaccines. Covaccines type of vaccine is an inactivated vaccine. Covaccine actually is an inactivated vaccine. So inactivated vaccine, what do you mean by inactivated vaccine? Vaccine here actually it is an inactivated COVID-19 vaccine. SARS-CoV-2 virus strain was isolated from an asymptomatic COVID-19 patient in the National Institute of Virology. Covaxin actually developed by the Bharat Biotech with inclusion with ICMR, Indian Council of Medical Research, and National Institute of Virology, right? And here actually virus in as an inactive pathogen. So virus do not cause the disease, but it will generate the immune response. That is the major goal of Covaxin. But now Covishield actually it's a viral vector. It is an engineered viral vector vaccine developed by the Oxford AstraZeneca COVID-19 vaccine employ more established technique, that is the viral vector vaccine. So vaccine used as a bioengineered version of a harmless common cold virus found in chimpanzee to instruct human cells to make antigen. Actually here the we use as a use the major form of adenoviral vector. Adenovirus actually it's a virus that cause common cold in chimpanzee, right? Now you can see that here how the Covishield works. Like in case of here, you can see that these are chimpanzee adenovirus. These are chimpanzee adenovirus, and these are modified form. These are modified, and these are CHADOX1. That's virus is unable to cause disease in human. That's a modified vector form. These modified vectors, these are unable to cause disease, and now these are modified vector, and now these are SARS-CoV-2. That's a virus that's contains single-stranded RNA. Now during this virus, the single-stranded RNA is converted into double-stranded DNA form by the cDNA. By genetic technique, we can convert from RNA to cDNA, and from double-stranded DNA, we know that genetic sequence of the SARS-CoV-2, and from this genetic sequence we can incorporate into the modified vector, and these modified vectors acts as a vaccine. So vaccine, that is Covishield, we are taking the CHADOX1 in COVID-19, the vaccine. So this vaccine, these are engineered form, injected into the human muscle, and then develop immune response which protect against future SARS-CoV-2 attack. Now second one is it's a only one forms of a vector is used here, adenovirus vector, and one of the important use is that here double-stranded DNA is used. Virus is single-stranded RNA, but in Covishield the double-stranded double-stranded DNA form is used, right? Now PIPZR and the Moderna vaccine, PIPZR and Moderna vaccine, actually it's a mRNA vaccine. It's a only single-stranded mRNA vaccine. The vaccine contains materials from the virus that cause COVID-19 that gives our cells instruct for how to make a harmless protein that is unique to the virus. Here actually in PIPZR, one of the more complex issues is the ultra cold storage temperature. Like here in case of PIPZR or Moderna vaccine, ultra cold storage is required, minus 70 degrees Celsius is required, because here RNA is used, mRNA actually used, and mRNA, we all know that mRNA is less stable than DNA form is highly stable. So for mRNA, minus 70 degrees Celsius is required for storage purpose. That is the major drawbacks of this vaccine, PIPZR and Moderna vaccine. And now another is the Sputnik-V. Sputnik-V, it's a Russian vaccine. Sputnik-V developed in a same way like in develop a Covishield, but here two different adenoviral vector is used. In case of Covishield, only one vector is used. Here Sputnik-V used two different adenoviral vector, AD26 and AD5. But it's now Indian companies develop, the Dr. Reddy's laboratory actually working for developing the Sputnik-V. So Sputnik-V works in a similar way to the Oxford AstraZeneca vaccine, that is Covishield vaccine. It is a combination of two different adenoviral vector, that is AD26 and AD5. Now it's what is this idea to use two different viral vector? Here is that idea is that using because all the vaccines have a two dose, the first dose and second dose. Now here the two different vector is used because the idea is that using two different formula boosts the immune system even more than using the same versions of the vaccine twice, and may give longer lasting protection. That is the major reason here using Sputnik-V, using two different adenoviral vectors. Now these are how Covishield works, and so thank you for watching.